The neurobiotic sense, the gut's newly recognized sensory capacity to detect microbial and nutrient cues and transmit them directly to the brain, is a fragile and finely tuned system. It relies on a diverse microbiome, specialized gut cells like neuropods, intact vagal nerve pathways, and a rich chemical dialogue between microbial metabolites and host receptors. Yet this intricate communication network is increasingly under threat from environmental disruptors that our biology never evolved to handle. Antibiotics, chronic stress, and industrial pollution may not only harm the microbes that mediate this sense, but actively distort or dampen the very signals our brain uses to interpret hunger, mood, and motivation. The result is a growing epidemic of metabolic, emotional, and behavioral dysregulation, rooted not just in psychology, but in the environment's assault on our internal sensory infrastructure. Antibiotics are perhaps the most direct and dramatic disruptors. While life-saving in acute infections, even a single course can decimate microbial diversity in the gut, wiping out entire strains that play essential roles in producing short-chain fatty acids, neurotransmitter precursors, and anti-inflammatory compounds. In animal models, post-antibiotic microbiomes lead to diminished vagal tone and altered expression of gut-brain signaling genes. In humans, early life antibiotic exposure has been linked to increased risk of obesity, ADHD, anxiety, and eating disorders. These effects may stem from the loss of microbial partners that normally help fine-tune neuropod signaling, appetite regulation, and emotional equilibrium. The damage isn't just about what's missing, it's also about what takes over. Antibiotics can allow opportunistic or pro-inflammatory microbes to dominate, producing distorted or excessive signals that mislead the brain. For example, certain species may overproduce lipopolysaccharides, LPS, or flagellin, molecules that activate immune receptors like TLR4 and TLR5 and trigger low-grade inflammation. This inflammation blunts satiety signals and sensitizes stress circuits in the brain, leading to a state of chronic discomfort that people often attempt to soothe with food, screen time, or other compensatory behaviors. Appetite becomes disconnected from energy needs, and mood becomes increasingly fragile. Chronic psychological stress is another potent disruptor of the neurobiotic sense. Stress hormones like cortisol alter the gut barrier, reduce microbial diversity, and weaken the responsiveness of neuropods and enteroendocrine cells. Stress also reduces vagal nerve activity, dimming the main communication line between gut and brain. With this two-way breakdown, the brain receives fewer calming or satiety signals from the gut, while the gut becomes more vulnerable to inflammation and microbial imbalance driven by the brain's distress. In this feedback loop, food choices shift toward high-calorie, low-fiber comfort foods that further starve beneficial microbes and fuel dysbiosis. This may help explain why stress is so strongly associated with binge eating, sugar cravings, and emotional eating patterns that feel uncontrollable. Even more insidious are environmental pollutants, microplastics, pesticides, heavy metals, and airborne toxins that increasingly contaminate our food, water, and air. Many of these chemicals have now been shown to alter the gut microbiome in ways that impair nutrient sensing, promote inflammation, and mimic hormonal signals. For example, bisphenol A, BPA, and phthalates, common in plastics, act as endocrine disruptors but also reshape microbial communities and reduce the gut's ability to produce serotonin and SCFAs. Particulate air pollution, PM2.5, has been linked to gut inflammation, microbial depletion, and behavioral changes in mice, including increased anxiety and impaired reward processing. These pollutants interfere with the biochemical language that neuropods and the vagus nerve rely on, leading to a sensory system that is overwhelmed, miscalibrated, or chronically misfiring. Importantly, these environmental stressors rarely occur in isolation. Many individuals, especially in urban, industrialized societies, are simultaneously exposed to all three: antibiotics in medicine and agriculture, psychological stress from modern life, and persistent exposure to environmental toxins. The cumulative effect may be a global desensitization of the neurobiotic sense, leaving people more vulnerable to metabolic diseases, mood disorders, and disordered eating, not because they lack discipline, but because the gut-brain circuits that once provided internal guidance have been compromised. Yet, even in this troubling landscape, hope remains. The neurobiotic sense is resilient, not immutable, but trainable. Diets rich in fermentable fiber, fermented foods, and polyphenol can help reestablish microbial diversity and restore healthy metabolite production. Mind-body practices like meditation and vagal nerve stimulation can enhance parasympathetic tone, improving gut-brain signal clarity. And emerging therapies, from precision probiotics to fecal microbiota transplants, may one day help rebuild the microbial foundation of sensory health. Ultimately, to preserve the sixth or perhaps seventh sense, we must begin to treat it as such, not as a passive digestive process, but as an active perceptual system that deserves protection from the same environmental harms we already know to be damaging to sight, hearing, and cognition. By acknowledging how deeply external disruptors can affect our internal wiring, we open the door to new interventions, not just to treat disease, but to restore the basic sensory fluency that makes us feel truly in tune with our bodies and our needs.